Hi, in this video you'll learn about the genes included in the GenXys pharmacogenetic panel. The completion of the Human Genome Project and the access to genome-wide association studies gave researchers and clinicians all over the world the tools required to implement pharmacogenetics into clinical practice. Most genes involved in drug metabolism or drug response can have changes or polymorphisms that affect how each person responds to a drug. For our pharmacogenetics panel, we selected only those genes that are backed up with the highest levels of evidence for clinical utility. This means that these genes have been shown to affect drug response in several studies. We follow a rigorous review process of peer-reviewed articles, systematic reviews, randomized controlled trials, and professional guidelines such as those published by the Clinical Pharmacogenetics Implementation Consortium or the Dutch Pharmacogenetics Working Group. Physicians, pharmacists, epidemiologists, and geneticists were involved in the curation process. At present, a total of 34 genes are included in the GenXys panel. The results obtained from testing these genes allow us to make a prediction or recommendation about the use of 116 drugs. Most of the genes that have an effect on drug response are involved in either their pharmacokinetics or pharmacodynamics. Pharmacokinetics analyzes the way that drugs move through the body, how they are absorbed, distributed, metabolized, and excreted. Pharmacodynamics analyzes the effect that a drug will have in the body. In most cases, this is determined by how drugs bind to their specific receptor, but other elements such as messengers that transmit the signal generated by a receptor will also have an effect. Highlighted in blue are some of the pharmacokinetic genes included in our test. The most relevant genes in this group are those that encode cytochrome P450 enzymes. These enzymes are responsible for the metabolism of 70 to 80% of all drugs in clinical use and are also a major source of genetic variability. Other genes such as ABCG2 and SLCO1B1 encode proteins that act as drug metabolite transporters, and changes in their functionality will also have a significant effect on the response to cholesterol-lowering medications. Highlighted in yellow are some of the genes involved in drug pharmacodynamics. Most of them encode receptors that bind drug metabolites. Therefore, genetic variations that influence the efficiency of that binding will have an effect on drug response. Other gene variations, such as those in B-cork-C1, are involved in the mechanism of action and have a more downstream effect. For each one of these genes, we evaluate up to 10 different variations, also known as single nucleotide polymorphisms, or SNPs. For CYP2A sits and CYP2D sits, we also assess the copy number variation, or CNV, which is essential in determining the effect of the genetic changes. The GenXys pharmacogenetic panel also evaluates the presence or absence of specific human leukocyte antigens, or HLAs, since certain variations pose a high risk of drug hypersensitivity. For more information about the SNPs, CNVs, and HLAs included in the GenXys pharmacogenetic panel, and how these can be used to optimize drug therapy, tune in to part 2 of this video.